So today, thanks again for joining this week's ASAP seminar. And we have our speaker today is Henry Sun, who's an associate research professor at the Desert Research Institute in Las Vegas, USA. And so Henry studied originally Antarctic microbiology for his PhD with Imre Friedman, and then later studied geomicrobiology during his postdoctoral work with Kenneth Nielsen. And before he took up his current position at Desert Research Institute, he was a staff scientist at the Jet Propulsion Laboratory, JPL, over in California. And so in the past few years, he's served on the editorial board of Astrobiology for 5 years up to 2018, and is currently an associate editor for the International Journal of Astrobiology. And so we're very glad that Henry today will be able to talk about some of his work regarding deamino acid metabolism in L life. And so with that, Henry, please go ahead.
Okay, well, thank you, Tony, for the introduction. So I'm very happy to be here, and from the previous speakers, it indicates to me that there's quite a bit of interest and enthusiasm in Asia, students in Asia and faculty in Asia Pacific region. So, and this is something that I actually have been wanting to do or have been trying to do for some years. Every time I go back to China, always go to universities and give lectures and try to sort of encourage students to think about life on Mars, particularly since China is getting into space exploration, space programs. So so this is very good to see so many people who are interested in the topic. So what I'm going to do is today is to really kind of introduce to you what I have been doing, share some of my experiences in astrobiology. As Tony introduced me, I didn't start in the field. I was studying, just like everybody else does. You study. There's only one biology to study, and that's biology on Earth. So we all trained in Earth biology. So the question is always when I talk to students, they feel they're excited to hear the topic, the search for life on Mars and in the universe, but they struggle with the question, how do I do astrobiology? Astrobiology seems such a far away goal. There's no connection to what you do on Earth in your lab. So I'm going to kind of walk you through the things that I did, and it didn't come all suddenly. It took some time. So really, I encourage you to continue to kind of think about things and see what you can do. The idea is really you don't quit what you're doing now, but rather to add a new perspective. So act local and think global has a new meaning now. So you have to think not just globally on Earth, but universally, like in the solar system, in the universe. So I think that's a perspective that will improve your work you do on Earth. So that's really sort of the message I want to convey today. So details, I don't know how much, because this is a mixed audience. So I'm trying to kind of distill everything to the essence and just give you a taste why astrobiology is exciting. Okay, so I just quickly introduce where I work, Desert Research Institute. A lot of people don't know what it is. So this is a institute that is part of the Nevada, the state's higher education system. But when the institute was created, it was put outside the university. So we are separate from the university. We're not part of the university, University of Nevada. We have our separate books, separate president. So everything, we're independent. So this gives us a lot of freedom to pursue the things we want to pursue. We don't have to teach. The state pays for the building, for the electricity, and they also pay for the executive positions. So the faculty had to come up with funding to support their own program. So we are called the Softly Money Institute. So that's where I am. So we do a lot of different things. There are faculty, we have about 200 or so PhD scientists and about another 200, 300 staff or support staff. So we do different things, all kinds of things, but mostly focusing on environmental science. So when the institute was created, this was 60 years ago, the state legislatures thought, since we are in the desert, desert research seemed to be a good name for it. But if you look at the portfolio that we do, what we do today, we do all kinds of things. There are people who do ice core research, people do marine biology. So the name really does not reflect the things we do at the institute level. So that's give you, and if you're visited with Las Vegas, so I'm in Vegas, we have two campuses, one in Vegas, one in Reno. So if you have an opportunity to be here, this is Las Vegas, as you know, has a lot of things to visit. So if you're here, look me up. So before I forget, so I want to announce next week's speaker, who is Dr. Matthew Lenaville from LC, who will be speaking about planetary diversity. So I'm looking forward to his talk. So just in the next few slides, so I'm going to start with amino acids, and that's the topic. I'm going to go into some detail, but at the end, I'm going to introduce a few recently funded projects mostly to sort of give you to show you where I'm thinking next, and also I'm inviting your collaboration, because from the previous talks, so there are a lot of chemists in the group. I'm really inviting you to think about some of the issues I'm talking about, and invite your input and perhaps collaboration. So now amino acids. So the next few slides, I'm going to give you some basics. This is a sort of crash course about amino acid chirality, and I'm not a chemist. So when when I was, I started as an ecologist. So this is not really my field. So the other message I want to convey is, don't let ignorance sort of stop you. Okay. So when we talk about amino acids, you heard probably the handiness of amino acids. That's because amino acids are chiral molecules. They are asymmetrical molecules because of this alpha carbon. On this alpha carbon, there are 4 different functional groups, such that there are 2 ways to attach these 4 functional groups to the carbon molecule. There's a left-handed way and right-handed way. So people often describe this as mirror images of each other. Okay, so this R group, as you know, this is the carboxyl group, H group, proton, amine, and R is the variable specific to each amino acids, the variable group. So the other way to think about this is like the 2 doors. You have a double door, you have a left-handed door, and one on the left and one on the right. They look the same, their composition is the same, but you cannot switch them, because if you try to put a left-handed door on the right hand, it won't work. But the compositions are the same. They are made of the same parts, except they are mirror images of each other. Now, the way we call them left-handed, right-handed is based on an optical property. The 2 enantiomers, when in pure forms, they will rotate polarized light in opposite directions, one to the left, one to the right, equal angles. Other than that property, the 2 enantiomers are identical physically and chemically. So this is very important. So such that abiotic synthesis, chemical synthesis, will produce equal amount of L and D enantiomers. We call it a racemic. It's a racemic mixture. So the the primordial soup, by definition, should be a racemic. So the origin of life, when life evolves today, so it has to break that mirror, has to choose one or the other. So that's a fundamental question in origin of life. That's something that's outside my field. So we know now, if you look at life on Earth, in all organisms, proteins and enzymes are composed of left-handed enantiomers, to the exclusion of D. So there's no D enantiomer are allowed in protein synthesis. So de novo synthesis of amino acids produces only left-handed form. So the factory of amino acid synthesis come out, give you only left-handed form. And the incorporation of amino acids into proteins is so exquisitely controlled. It does not allow D to be incorporated. If you try to force D into the protein synthesis, it can't be done. So life, we said, is homochiral because it's based on only one on the left-handed amino acids. So the emergence of homochirality is generally the emergence of life. So we often refer to this condition as homochirality, and life on Earth is left-handed. Okay, you often hear this. People say that we are left-handed life form. So to simplify, I call this left L life. Now, if you go to another planet where you have independent origin of life, you might have L life or D life. So we don't, a priori, we don't know what you will find. Okay, so this is the background, and this is very simple. From here, you can do research, and that's where I started. So, there's one more fact. So the other property of amino acids is that it is not stable. So this left-handed form, over time, will racemize. So I call this sort of unstable building block. It's not a perfect building block. So it doesn't stay that way. The left-handed amino acids does not stay left-handed, because over time, once it's made, the proton, so the proton on the left-handed amino acids will come off and give you an intermediate, which is a planar, because you lose chirality. Now the three remaining functional groups are no longer constricted. They are rotating freely, okay? Now this intermediate is not stable. Immediately the proton, it will pick up a proton, and this proton can attach either from left-handed side or right-handed side, equal 50-50% chance. So you have a 50-50% chance of producing, having the new amino acids, left-handed or right-handed. So it doesn't matter whether you start with an L or D. Over time, given time, it will go through this racemization process, and in the end, it end up with the equal amount of L and D. So that's, we call it racemic equilibrium, okay? And for this reason, we cannot live forever, because you cannot stop amino acid racemization. As long as you have the temperature is not at absolute zero, this reaction will continue to occur slowly. Okay, so in 100 years, you're not going to have a lot of racemization, but in your eyeball, in your tissues that don't turn over, will accumulate some amount of D, and this is known. Okay. Now, so I'm going to start talking about the history of astrobiology of Mars. So the first mission, the first life detection mission, this is called the Viking mission. So there were two landers, the landing missions. There were two landers landed on Mars, and on each lander, these are identical instruments. They landed on the opposite side of the planet, and each carries several instruments designed to look for evidence of life. This is a biological mission. Okay? Now, one of the instruments is called the Labeled Release Experiment, or LR, designed by this gentleman called Gilbert Levin. So I was fortunate, had the fortune to meet with him and discuss with him. So what he did here is that he took the idea. This idea is well established in Earth biology. That is, soil microbiologists do this all the time. They measure soil respiration. This is essentially an instrument that automatically measure or assay soil respiration. So the idea is now to do this on Mars, you label your nutrients. So this is a heterotrophic broth, okay? The things that we grow bacteria in on Earth. So now each of this compound is labeled with C14, and such so that this broth is radioactive. So you add to this vessel a sample of Martian soil, and then into it you add your nutrient. Okay, so the nutrient is labeled, and then if there are microorganisms, they will chew on the sugar and amino acids and will release the carbon. Some portion of the carbon is released as a waste product, like we do when you eat bread in the morning and you're taking in oxygen and you exhaling CO2. That carbon in the CO2 came from the bread you ate. The same idea here, except that now here, this carbon is labeled. So the label now will emerge in the overhead, will appear in the overhead space. And you have a Geiger counter that counts the radiation. You're not analyzing CO2, you're counting the radiation. It's a very elegant design, it's very simple. So he tried this on Earth, I don't know, 1,000 times, many years, with all kinds of samples, and it worked reliably. Reliable in the sense that if you autoclave the soil, that signal will disappear. Okay, so that's all set up and everything worked, and it went to Mars. I'm sorry, I'm forward. Okay, so this is the result. When you inject upon injection of the nutrient, radiation counter goes like crazy, going up. So gases, the label now appears in the overhead space, okay, very rapidly. So this is over time in sols, days, essentially days. On Mars is called sols, okay. So within a few hours, or one or more compounds in the nutrients were degraded very rapidly. Now, if you autoclave, so this is a control. So the lander has an onboard oven. If you cook the soil to 160 degrees Celsius, so this is basically sterilizing temperature, and you do repeat the experiment, and nothing, flat. So this is exactly what Earth soil will behave, and mostly nearly all Earth soils contain bacteria. So there's activity, there's a release of the label. If you don't autoclave it with fresh samples, pristine samples, if you autoclave it or sterilize it, the activity disappears. Okay. Now I'm going to summarize what people think of this experiment. This experiment apparently is not what people remember it, because if you ask Gil, he thinks that you may have read his papers. To this day, he thinks he discovered life on Mars. Everybody else is just too dumb to appreciate his finding. Okay, so everybody else, almost everybody else, thinks there's a problem. The problem comes from this. There's another instrument on board the lander, the Viking lander. Is a pyrolysis GCMS. I'm sorry, this is a typo, which has a detection limit of ppb, parts per billion. Well, this is also a typo. Now that instrument detected no indigenous organic carbon. So what does that mean? If you don't have carbon, you can't have bacteria, okay? So Viking detected apparent bacterial activity because the bacteria, the LR response is consistent with pre-launch criteria for life detection, okay? But no bacteria. So to put it another way, light is on, but nobody's home. So this is the dilemma, okay? To reconcile the results from these two instruments, it's generally thought that there is an inorganic or one or more inorganic oxidants that is highly reactive, and that's what caused the reaction. Things like superoxide radical, OH radical, hydrogen peroxide, things that can be produced by UV radiation. Somehow is stabilizing the soil, and when you add the LR nutrient, boom, there's a reaction. Okay, so this is the conclusion that the Viking team made as a group. But if you ask Gil Levin, he thinks there's life. Okay, so I started to okay. There's another story after this. So fast forward about 20, 30 years. I'm looking at the time. So Chris McKay and my former major professor, and this was started quite far back when I was in graduate school. They started going to Atacama. The reason Atacama is interesting because Atacama, there's a part of Atacama that is very dry, essentially rainless. As a result, no primary protection, no plants could exist. Even algae, lichens disappeared. They couldn't exist. So this became a very interesting place. And Emery Friedman and Chris McKay recognized that this may be a place that we can understand limits of life, dry limits of life. And so they started working quite some time ago. So to summarize in the meantime, I think Emery Friedman passed away, so Chris McKay continued to work on this. So this is what they did, and you may have heard about Atacama having Mars-like soil and such, and a lot of people are going to Atacama to test concepts, life detection instruments, and this is the paper that tells you why people are going to Atacama. So what Chris and his colleague did is essentially repeated the LR in Atacama, but instead of combining, I kind of skipped this when I was talking the LR nutrients. The LR nutrient, because life elsewhere may not use the same enantiomer as life on Earth. So what they did is they combined DNL into the same, into a mixture, okay? So it doesn't matter if what Mars is like a buffet. You can pick and choose. I don't care. But if somebody is alive and eats either LRD behind the LR. So what Chris McKay did here, his colleague did here, is that to separate the isomers into two different tubes. So the biotic soup that contains D-glucose and L-alanine. Biotic, they explicitly stating that no organism on Earth could utilize L-glucose or D-alanine. Okay, so when the incubated Atacama soils in these two test tubes over a period of weeks, then they found this. So in the abiotic soup, when you mix the soil with abiotic soup, there's a very small amount of reaction, and it doesn't matter where the soil is coming from. So over here, the soil are coming to this. This is a transect going from wet. Wet is like not like the Amazon forest, but wet there are like a few plants. So there's still some productivity here. Going toward the equator, became very dry. Eventually, life, as we know it, disappeared. There are not even bacteria in the soil. No DNA could be recovered from soil here. So we can call this Earth soil and Mars soil. Okay. So somewhere there's a dry limit that is crossed. Now, when he did this incubation, in the abiotic test tube, there's a reaction, and the reaction does not vary across this transect. It's constant. It's very little bit constant. Now, if you incubate the soil, the same soil, replicates in the biotic soup. So the way they plotted it is a little funny here. So I just called because they added this up. So this increase in the signal is coming from the abiotic reaction, the biotic reaction. So as you go this way, the signal from the biotic test tube increases because there are bacteria. The bacteria now are chewing on the D-glucose and L-alanine. So this is well known because that's the nutrients that we have in our test tubes when we grow bacteria. So this is expected. So this now, if you look at so this is a biological soil, and the signal is dominated by the biotic response. Here, what he called Mars-like soil degrades the two soups in equal quantities. So this is a characteristic of a chemical reaction. Okay, so he called this abiotic. This is not so implying that there's some inorganic oxygen in the soil that is just as on Mars, that is reacting with the two enantiomers equally. Okay, so this is where the Mars-like description came from the equal reaction with the two set of isomers. Move forward. Okay. So this is the conclusion. There's Mars-like soil in the Atacama, Chile, and dry limits. So this was 2003. Now, so I started looking at these assumptions. The assumption that no bacteria on Earth or no microbes on Earth could utilize L-glucose and D-alanine. So we worked with the sugars, and glucose indeed is the case. So every organism we look at, whether it's E. coli, whether it's bacteria or archaea or eukaryotes, when you give them D and L glucose as a carbon source, they grow only in the D glucose. So D glucose rapidly disappears from the media. When you incubate them in L glucose, they will sit there and the glucose doesn't go anywhere. So the bacteria would starve to death floating in sugar, because this chirality is not available to the organism. So the chirality presents a barrier to metabolism. Now, we also did it with the amino acids. So that's where we were surprised. So if you add, now again, that two different tubes, which has the soil from Atacama. This is biological soil, by the way. So the soil contains bacteria. When you add L amino acids to the soil in the aqueous solution. Now, let me find my cursor. So you see, just focus on here. Alanine to L to the open circle is L, and the filled circle is D. D and L are consumed equally, removed equally at the equal rates from the medium. Because we didn't know where they go, but we assume they went into the bacteria because they don't have anywhere else to go. The other amino acids, L goes very fast, disappears very fast, but the D also disappears over time. It takes more time for the bacteria to start utilizing them, but eventually they will start utilizing them. So you say, well, that's kind of weird because we know that proteins and enzymes inside the cell are made of L only. Where did the D go? We can understand where L goes, right? Because it went into protein synthesis. But where did the D go? So kind of start really kind of pulling on the question. So we follow this up. So this is what we think is going on. So all bacteria contain amino acid racemases. I'll explain a little bit later why they need racemase. So this is a special enzyme that catalyzes the racemation reaction. So you just flip, amino acids flip left and right naturally, thermally, but very slowly. With this enzyme, it happens much faster. In minutes, if you start with a sample of L amino acids, give 20 minutes, you become racemic. Okay, that's what the enzyme does. So all bacteria carry racemases. Okay, now when you give them L amino acids, of course, L, we know what happens. It imported, and then it went into protein synthesis. Okay, it served as a nitrogen source. Now, usually they have to take the amino acids apart to make other amino acids. Okay, so if you give them one amino acid, from there they can make 20. Right? Now, if you give them D, if you deny L and give them D instead, this is what we think it happened. So permease is not sensitive to chirality. So when it can import L and D. Now, because L is not here and D comes in, because there's racemase here. So D then will convert to L. Because L is constantly removed, then D will flow this way, even though this is a reversible reaction. So I call it reverse racemization, okay? And then it goes into L amino acids, okay? So this is what happens, I think, why in the sample we observe the disappearance of D amino acids from the media, okay? Now, so some microbiology, some molecular biology actually found, and this is kind of tied into this, that the two enzymes that are responsible for L amino acids catabolism or utilization, the dehydrogenase, so this is dehydrogenase, and permease, are coupled, are grouped together, if you will, with the racemase. This is the L-alanine racemase, and put this under the control of the operon. So for non-biologists, if the operon is a way of coordinated expression of several enzymes that are needed to perform a single function. So you want to upregulate or downregulate all the enzymes or turn them on and off the same way at the same time. So you don't waste energy. Because if one enzyme is expressed, the others are not, they're not doing any good. So this is these three enzymes are controlled under operon, a single operon. So this is well established. And this operon, interestingly enough, is induced by L amino acids. So not by D. So when the bacteria are using L amino acids, racemase is expressed along with the other two. But it's not used at that time because you don't need the racemase to do anything when L is is available, okay? But once the L runs out, now racemase is kicking in action because it needs the racemase, this racemization in order to start utilizing D, if D is available, okay? And you could ask, why don't you wait until you finish using the L and then start using D? If you wait, you run into a problem, into a dilemma, because if you start to synthesize racemase, then you can't, because all enzymes are made of left-handed amino acids. L amino acid is no longer available, so you cannot start making racemase after you depleted all your L. So that's the reason. So the bacteria are very smart. So this evolution kind of push for this mechanism. So we did the experiment. We, instead of adding the two enantiomers in separate tubes, we add them together. So we have a way of monitoring the two isomers with a special procedure. We can still distinguish the L from D. If we add them together, and this is what happens. So just focus on the Atacama soil. L is always used first. But once L falls below a certain threshold, D start to come in. So D, the utilization of D doesn't have to wait. Once L is gone, D utilization starts. So it's a smooth transition, okay? And then when you say, why do you need to be smooth? Because if you sit in D, pure D amino acids, it's a very bad idea because D amino acids is toxic. Okay, so D is toxic to our life. So you have to learn how to get rid of it. So implications. So very small finding, but you can think about what does it mean. So for Mars, it's clear because the assumption that no organisms on Earth could utilize L-glucose and D-alanine clearly not completely correct. For glucose is yes, but D-alanine is not true because bacteria have the enzymes, the repertoires to use D just as well as L-alanine. So this means if you do live detection on Earth, on Mars, and go back to Mars, redo the LR experiments the way that Chris McKay did in the Atacama, then it's better to keep glucose and alanine separate. Don't mix the two, okay? So one can also think about life on Earth. So why, what does it mean? So I'm gonna speculate here. So this is a statement I'm making here, that the emergence of amino acid racemases in bacteria was a very important event in early evolution on this planet. Because when life arose from the primordial soup, immediately our life is swimming in D amino acids, and subsequently because due to racemization. So life always had to deal with toxic D amino acids in the environment. In the beginning, there was no efficient way. There's environmental catalyst, protoenzymes, but when the racemases evolved, it became a very efficient way of recycling D amino acids. So efficient that the bacteria started competing for it. Essentially, create a D-free environment. So if you sample the environment today, there's no D, free amino acids, free form of D amino acids. Okay. So actually, I was jumping. Moving forward, not backward. So I was actually getting ahead of myself. To implication number one, because bacteria now have racemases can detoxify D amino acids, they now can make use of D, because they can now start making structures that are made of D amino acids. Because when you mix D and L and make a peptide, it's very hard to degrade. So hence, the peptide glycan cell wall, this is a new design of cell wall emerged. So here the enzyme, the racemase take L and make it into D, and D is incorporated into this cell wall structure. So most the bacteriologists would know that the cell wall, the peptide glycan, so this is the cell wall structure this is the lipid bilayer, and this is the peptide glycan. This is like a multi-layered cross-linked fishnets. It's a three-dimensional macromolecule. It's very physically complex, chemically complex. It's very hard to degrade. Okay, it's very stable. So if racemase didn't evolve, you couldn't make peptide glycans. Indeed, if you knock out the gene for racemases, then it's a lethal deletion. Bacteria can no longer grow. Okay, so the peptide that contain D is in this what is called cross. This is so if you take one slice of the wall, and this is what look at. There's a glycan backbone, and these are the peptides cross-linking peptides. Okay, all these short sticks. This stick is not cross-linked yet. These are cross-linked. So this is if you draw out the molecule. So this is the glycan chain, and this is the tetrapeptides, the cross-linking peptides. Okay? The peptide is a mixed chirality, has L amino acids and D and unusual amino acids. It's very complex. So this structure is very hard to degrade. So common protein enzymes cannot dissolve this. Okay? So the racemase it was paved the way for this structure. Very much like oxygen. When oxygen evolved, organisms first have to figure out a way to protect themselves from oxygen. And then you can start breathing oxygen. Okay? So detoxification always precedes utilization. So the second implication is that, I'm sorry, this is already said, already discussed this, is that once the bacteria learn how to compete for D amino acids, D amino acids, free D amino acids, there's always combined D amino acids as part of peptide. But free amino acids, as soon as the peptide is broken up into free amino acids, D and L are immediately utilized by bacteria. So there's no longer free D amino acids, free D available in the environment. So in other words, all earthly environments are D amino acids free. This, I think, paved the way for the emergence of complex eukaryotes. Without this condition, eukaryotes cannot evolve. So you can do simple experiment to test this idea. In other words, if you grew plants and in germ-free conditions without bacteria, and then give them free D amino acids, they should suffer. Okay, so we started actually doing this experiment. So this is I don't know if my student in China is in the audience. So this is Zhang Ling's work. These are green beans, bean plants. You grew them in plant medium. Just to show you that this is MS medium, they grew fine. But if you add D-aminoalanine to it, it suffers greatly. Okay, if you add bacteria as well as D amino acid, it improves. So this is Bacillus. Bacillus is not very good at depleting amino acids. So the rescue effect is not great. So we're thinking of trying other bacteria to really that we think would be able to rescue and restore the growth. So bacteria are really do us a service that is to clean up the environment of D amino acids. Okay. So some biologists might actually start a question. So I'm kind of trying to, because eukaryotes, plants do have what's an enzyme called D amino acids specific oxidases. So this enzyme binds D amino acids, but not binds L amino acids. So it's specific, it's chiral specific. So these amino acids, I think, serve sort of an internal patrol. So it works in your tissue, inside your cell. If there's some, any D amino acid appears in your tissue because you live for a long time, right? There may be D amino acids appearing in your blood tissue, in your bloodstream. So this will take care of it. This will take care of it. So this is a second sort of defense mechanism against amino acids. It's very specific, but not enough, but not sufficient to defend against a high amount of D amino acids, as we showed in the plant experiment, okay? So I'm approaching, I'm looking at the time here. So I'm gonna finish in the next couple slides and then end there. So I have more slides than I can finish, but that's okay. So I'm gonna zoom in on this reaction, the abiotic reaction. So what is in the Atacama soil that is degrading the L-glucose and D amino acids, okay? So trying to kind of tackle it from a chemical perspective. I just quickly show some experiment here. So we collected soils from Atacama, and we were able to recover. So this is in collaboration with a chemist in Greece. We're able to measure superoxide radicals in the soil, and I want to show you here. So I want to show you sometimes can do things, work on Mars experiment on your roof. So being in Las Vegas, this experiment actually was carried out on the roof of my building in the summer. So you go up or outside in the balcony, and you spread a layer of soil on tin foil, and every hour or so you collect it and you bring it back to the lab and do an assay. So you can study the production of superoxide radical formation in the soil. Okay, so we were able to show that we think the same chemistry that was suspected to be happening on Mars is happening on Earth, in Las Vegas, in deserts, as long as the soil is dried and exposed to UV. Okay, here of course it's not UVC, but it's just UVA and UVB on Earth. But nevertheless, the reaction does happen. We were able to detect it. So this is our sort of understanding, sort of illustrating in this diagram. The UV is interacting with the mineral, produce radicals, superoxide radicals, and in the presence of iron, it can produce, it goes through this Fenton reaction. Chemists would be familiar with this, and giving OH radicals that is very, very reactive and drive all kinds of redox reactions, oxidizing organics, producing NOx, nitrogen oxides. So there's a lot of chemical reaction going on due to this UV radiation. So this is so I will, let me look at time here. So I talked earlier about peptidoglycans containing D amino acids, okay? So I want to show you another observation I made that as on Earth, we don't usually think about. So if you ask a chemist, amino acid chemist, and say, how do we look for life on another planet? He would say, it's very easy. You look at the chirality. You collect the soil, you extract amino acids, and you quantify L and D. If you find equal amount of D and L, it's prebiotic. There's no life there, okay? Or life was there before and has been, was long gone. So you're looking at a extinct life. But if you have life, living organisms, then you will find L in great excess of D or the other way around, D in great excess of L. So if you find fossils that are relatively young, there are a few million years. On Mars, it will take longer. So resonation is a function of temperature. So this excess could preserve longer before it becomes racemic. So if you find unequal amount or comparable but still unequal at L and D, then you say, well, there's a fossil. This is fossil. Okay. So I want to show you one analysis that shows that this is not always the case. Not always the case. I'll just skip here. So this is something that Jeff Beta did. This was a long time ago. He collected seawater. So you filter the water, organisms out. These are dissolved organic matter. You analyze the alanine, D and L, as a function of depth in the ocean, all the way to deep ocean. In the surface, because there's a lot, these are freshly produced amino acids or peptides, there's quite a bit of L, but already D is enriched. There's an enrichment of D. When you go to in the deep ocean, it's almost racemic. So this is quite remarkable. So the conclusion here, I think, and chemists should look into this, that the seawater, I think, and other people are saying this, seawater is a bacterial graveyard. It's an organic soup of peptide glycan remnants. And these remnants, I think, is the long-term nitrogen source for bacteria and everything else. Okay. So what is more remarkable, if you think about astrobiology, if aliens come to Earth, landing in the ocean, and look and using the understanding, like I said in the last slide, using chirality, amino acid chirality to look for life, you would think there was no extant life in the ocean. Life existed some time ago. That's probably millions of years ago, but not today. So that's quite a remarkable thing. So this, I think chemists, somebody with an analytical background, should really look into this. So just quickly go over a few slides. So this connects to my early work when I was in graduate school. I was working in Antarctica, and so this is show you where I was. So the dry valleys. So most of Antarctica is under a big glacier. So this is the field where I was doing field work. Okay, sandstone with what we were studying. And this is what looks, if you get close to one of those, you see this exfoliation. The crust is popping off, okay? Not rapidly, but not observable, but over time, it exfoliates, it weathers. This is caused by activity, biological activity underneath the surface. I think Maggie talked about this in her talk. So this is a Indolithic community underneath the surface. This is what is causing the weathering. So this bar is about 10 centimeters. This is about 5 centimeters or 5 millimeters from the surface. This is surface. So I wouldn't go into the detail about the ecology or biology, but to just show you one analysis. This was discovered quite a while ago by Emery Friedman. What I did here is I cleave the sample 1 sort of millimeter at a time, 1 grain layer at a time, and then extracted amino acids. So that includes living cells and dead cells and everything in it, and then did amino acid analysis. So some amino acids, so this is depth. So this gray zone is the biological, the colonized zone. Below that, there's no organisms. So these are all dead or fragments of organic matter. So in the colonized zone, near the surface, it's more productive. So the biomass is higher near the surface. This measurement does not indicate the quantity of biomass. Quantity of biomass should be going like this, high near the surface and taper off near the bottom. But if you look at the amino acid ratio, so this is what is ratio of D amino acids as a function of depth. So some amino acids, the ratio is 0. Tell you there's no D there. All the amino acids are in the form of L. But a few, as you go deeper into the rock, the ratio of D is increasing. And so this does not conform to the picture I started with, that a living community should contain L only. Not true. And the reason we understand why, because peptidoglycans are accumulating, fragments of peptidoglycans are accumulating in the community as it's reaching down into the rock, okay? I think I went here. I think I almost approached the hour, the end of the hour. I'll maybe stop here and take questions.
Okay, thank you very much, Henry. So for the audience, this is your time now. You please feel free to ask questions. If you have any questions, please type in the chat box, either type in the question itself or just type in that you have a question. And then I'll get to that. So there's already a few questions right now. I'll start from the top. And again, if you have any other questions, please feel free to write it in the chat anytime. So Maggie and Philip ask if they heard you correctly. It was explained that the racemase evolved such that life could begin to use D amino acids, such as incorporation of DA into the cell wall. Is there any information about the time of the emergence of the cell wall and racemase?
You mean, I guess the question is like, at what point, right?
Yeah.
How long ago? That's a great question. I don't think anybody talked about it. So if you look at it, so in archaea has a cell wall structure that's very comparable to peptidoglycan. It's called a pseudomurine or pseudopeptidoglycan. The structure are very comparable. And so the relationship between that and true peptidoglycan is quite interesting. And at what point it emerged, I have no idea.
Okay, great. So the next question is from Andre. He asks, is it possible to have, for example, L life and D life existing side by side and benefit from being more competitive in using their respective isomer? If so, why isn't this the case on our planet? And was this the result of random emergence of one type of life form quickly evolving to occupy both niches?
Yeah, that's a great question. Actually, a lot of people, Paul Davis, the Arizona State, speculated about this, what he called a shadow biosphere. Could you have a second genesis on the same planet in a way that is not that you have it could be just under your nose and you wouldn't know because it's utilizing different mechanisms like D versus L. I don't think it's possible because once L life emerged, L life have to quickly figure out how to get rid of D from the environment. So the condition for origins very quickly disappeared because life is ready is spreads, right? It's not confined. Once life emerged, sort of like the queen of in the beehive. The first one that emerged, it killed everybody else. So I think once L life emerges, then it will eliminate all the amino acids D and L. So D life couldn't have emerged on the same planet.
Yeah, and just I want to add one small point. So I think this is also an interesting question with respect to synthetic biology or kind of protein engineering, where people have been trying to engineer these mirror image proteins or mirror image kind of biopolymers. And there's actually one demonstration from a kind of a prebiotic chemistry group showing that ribozymes can actually be cross-chiral and they can work together with each other. But that's just like one example of a very kind of in vitro engineered system that could show such response.
Actually, there's a, you're right. I'm familiar with that work too. There's another study that was done, I think, quite a while ago by a chemist at Caltech and who took an enzyme, I think the lysozyme, from the HIV virus. And I think because it's a relatively small enzyme, so it sequenced the protein and then synthesized in the lab with D amino acids, all D. So the karate is completely flipped. And then did some studies. So the D enzyme, if you will, the D enzyme no longer recognize the original substrate. The substrate, which is a peptide, also have to be flipped and to be able to interact with the D enzyme. So D life, in other words, at least on this, demonstration that D life can exist. But again, I think it has to be on a different planet, not the same planet.
Great. So the next question is from Simon. So he wants to know, in what way are D amino acids toxic to life? So you showed a very compelling diagram of plant growth with D-alanine. And do you know what exactly is the mechanism of this inhibition?
That's a great question. So there's a couple of groups. There's some old literature that was looking into it. Today, there's a group in Europe that is looking at it. As far as I know, this is a subject nobody's looking at. Why D is toxic? What happened to it? Apparently, because plants don't have a need for organic nitrogen, right? It typically does not prefer nitrate, ammonia, in organic nitrogen. So why would you have a amino acids transporter in your roots, your root head, which is quite bizarre. So it makes the plant vulnerable. But because I think so that I don't know, to be honest. Apparently the roots can take up amino acids, but for what reason do they do it? And once you get in, so sometimes people have found that if L amino acid getting into the plant, it can also create problems.
Mm-hmm.
So this is an area. So that's, I guess, the message. This is the questions are very good. If you're a plant person, you can do astrobiology.
Great. So Joe would like to know about the cryptobiotic crusts in the Mojave. Do you have any idea whether they're racemic or not?
Well, so this is a topic that I want to briefly mention, but I didn't have time. So we got a new grant where we are looking at the UV screening. So a lot of people have done research using spores, bacilli-induced spores, to study the effect of UVC on Mars. The exposure with the soil crust from the Mojave. So this black surface is soil crust. This is a mat of cyanobacteria, lichens. Now they don't look green because they are covered on their top. So let me show you quickly. This is soil crust. They look black, right? They contain chlorophyll, but they look black. The black color is from the UV the sunscreen compound, screening compound. That's what it is, a cross section. The top is black. There's so much sunscreen that it looks black from the surface. You can supply UVC to them. It will take hours, sometimes days, and it is still alive. So I'm starting to, using these organisms as a way of really kind of challenge the notion that nothing could live on the surface of Mars. Now, I'm not saying that things could exist on the surface of Mars. What I'm saying is if other conditions are suitable, if temperature is there, if water is there, nutrients is there, then things could survive. So UVC is a very easy problem to deal with, because once you make the sunscreen, then the UV is blocked, right? It doesn't even reach the cells.
Okay, so the next question is from Giovanni and so he asks whether D-amino acid consumption only occurs in microorganisms that live in extreme environments, or are there also demonstrations of kind of more less extreme environment microorganisms?
So that's a good question. Bacteria were the first organism that started utilizing D because they invented the racemases. So the peptidoglycan is the consequence of that invention, enabled by the invention. Now, today, because the peptidoglycan synthesis, it increased the amount of D in the environment, okay? Because bacteria accumulates. The peptidoglycan fragments accumulate in the environment, in the ocean, sediment, soils, everywhere. Everywhere you have bacteria, you have that. So to answer the question, all bacteria contain peptidoglycan. That's the basic characteristic of bacteria. Cyanobacteria, bacteria all have peptidoglycan. Now you can ask, so interestingly, other bacteria, other organisms start to steal, if you will, through horizontal gene transfer. So racemase genes is now getting into archaea, getting into plants, getting into fungi. But some of in some of the organisms, the enzyme used to make peptides that contain D amino acids. But in some cases, they are using the enzyme to catalyze, to compete with bacteria for D amino acids as a nutrient. So this trick is now being stolen, if you will, by other players. But the bacteria are the best at these two things.
Okay, great, thank you. And then Ram Ganesh has two questions, so I'll do them separately. Yes, there's three questions, so I'll do them separately. So the first question is, outside of internal racemization within bacteria, have you identified any external factors that can possibly influence the naturally available DAA racemization? Do external factors such as pressure in the deep sea or high temperatures in desert soil have some influence on the racemization?
Oh, that's a great question. Chemist, somebody's a chemist in the audience, should really think about this. So Jeff Beda, so I was citing his work. So he did a lot of these experiments. So extreme pH, either extreme acidic or basic, would speed up the racemization rate. Other than that, temperature, if you raise temperature, hot springs, racemization go much faster, okay? Other than that, we don't know. Now, from an evolutionary standpoint, that because now we have an enzyme, we have racemases. I would think there have to be some protoenzymes that are better than the acid versus hydroxide versus temperature. That was the bridge, from to the enzyme that bridge this sort of physical environment, physically sort of catalyzed reactions versus real enzymes. There has to be protoenzymes, I think.
Okay, so then the next question is if, for example, if one is using D amino acids for enrichment processes for deep sea microbes or any other kind of sample, what would be a good mixture of kind of D amino acids to start with, for example, would a mixture of canonical and non-canonical D amino acids in separate experiments work, or would mixing both kind of canonical and non-canonicals in a single experiment?
You know, so we have been doing some of the experiment with soil bacteria. We are, this picture is very messy. It's not an easy experiment. I actually don't know. I would start with the common amino acids like a D-alanine, D-glutamic acid, those common ones, because they are present in the environment in large quantities. Usually, if a bacterium is using D, can use D to grow, those are the ones they will use. So these are a good amino acids to start with. And that's true that some amino acids also are non-conical. And those, like, if you think about meteorites, right? So one could also look at the amino acids from meteorites and say, would Earth bacteria be able to utilize those? So those would be good experiments to do too.
And then I think this is the last question. So this is kind of going back to that first Atacama Desert experiment. Is there any specific reason for choosing those specific 4 D amino acids, for example, abundance or something? And then kind of why we're not, or what was the reason why those 4 were chosen and not more, like, 8 or 12 or all 20?
Oh, I'm sorry.
So why exactly were the 4 D amino acids in the first Atacama Desert experiment, why were those the ones that were chosen to be assayed rather than other ones or kind of a larger pool of amino acids?
Oh, I see. Because why did I choose 4? So this is due to an analytical constraint. So some amino acids are not soluble in water. They have a low solubility. You cannot have enough in the solution. So there are some practical issues. So we just started with 4. You could try 10. There's no particular reason. So some of the amino acids are if they are non-polar, they are they don't dissolve very well. So you cannot really study them very easily.
Okay, great. So if anyone has any other questions, please say so now. If not, let's thank the speaker again for a very interesting talk. Please feel free to stay on this channel as long as you want, if you want to speak a bit more after the talk. And then hope that everyone tunes in next week for the next edition, which will kind of move a bit towards the planetary science side, and there will be a bit about kind of comparative planetology. And so again, thank everyone for coming, and hope to see you again next week.